because I did give you a pretty small data set to look at, and it's a pretty conserved protein. But, you know, that's the kind of thing I'm gonna look for in your project, and the kind of responses I expect on the exams. We will have a little bit about tree building techniques. Okay, so again, if you're running into problems, then you can, Tuesday will be a great day to look into that. By Tuesday, you definitely should have all your sequences into Mega, because then you can, in that 3 hours, you should be able to whip through the rest of the lab, okay? So that'll be your goal. You might even get that done today before class is out, if all goes well. And I double checked all those numbers because some of the accession numbers have changed, so they should all be good, because I just redid them all last week. Any questions or concerns? Okay, good. All right, so I'm not gonna talk about anything but the bootstrapping from the last time, because I kind of wanted to not talk too much today, so that you can get going. So again, we have work days on the 20th, 29th, and the 4th. The 27th will be a speaker, and the rest of the time is a work day. So there's no lab associated with that. So, and then on December 4th, that's a Tuesday, then we only have two more class sessions. We have Thursday and the following Tuesday. So that Thursday, I'll be talking about genomes and also gene prediction programs, which is really I think it's chapter 9, because I actually haven't talked about that yet. So it'll be kind of a long lecture on the 6th. And then on the, what we do on the last day is, I'll probably try to get these done over Thanksgiving, if not a little before, because I have you read papers and you, that are recent papers using bioinformatics tools. And I try to pick a different, you know, in the past I usually do at least one plant one and one animal system one, so that you can kind of see how people are using bioinformatics currently. And usually what I do is this, I've done it different ways. One way is I made everybody read every paper. Another way is I break you into small groups and each person reads a paper and kind of jigsaw it. And then you talk in your groups, and then we talk as a whole group about that. And so those kinds of things will, and then you're held kind of accountable for it, because I do ask a question or two on the midterm. I don't call it a final because it's not cumulative. Okay, does that sound good? I can't believe that we're getting close to the end here. So, all right. So that's where we're at. Well, actually, lab 9 should have already been in. So you're going to start lab 10 today. Okay. All right. So bootstrap analysis. Again, I just want to, because I feel like I went through this super fast, but it's a resampling method to see if you can get the same tree. So again, I talked you through how to do it with columns, where you have, if you have a 100 nucleotide block, then you have all of these columns all the way up to 100, right? And this is based off of your lineup of sequence A, B, C, D, E, etc. And so when you have a sequence, you've got let's just say that, we're gonna say this, that these are all the same, you know, that you have your start site might be one of your first set of things. But it also could be that this sequence started here, so then there's a gap here. So when they resample, they take this as it is, so it's dependent on your MSA being good. And so in that MSA, there's a gap here, which is fine. And then it takes makes a new block, which I guess I could put in green, of 1, 2, 3, 4, okay, so all the way to 100. But in that block, okay, so this is a set, they're gonna put column, they're gonna take the column from the original sequence, which I'll put in purple, maybe column 60, and then they're gonna take column 45, and then they're gonna take column 60 again, and then they're gonna take column 2, you know, etc. And then they just put them in a column here. They don't do any kind of alignment. It's already aligned column by column. And what they do is they then rerun the tree. So in rerunning the tree, if they get the same tree, the same branches, then those branches are getting a 1. And then the second time they do, it gets a 2. And then the third time, maybe it switches branches, so there's a couple branches that won't get a score. In other words, if what we're looking for is in resampling, and then they resample every time, right? So it's they do it 500 times or 1,000 times, iterations. And if they get the same tree, then the same clade of 2 sequences, then it's gonna be 100. Let's say 70% of the time they get the same, then it'll have a 70. If it's 20%, then usually they leave it blank, which means there's not a whole lot of support for that tree. Yeah, I mean for that clade.
So, does the program only look at it one column at a time? Because then if you're moving around the column is gonna stay the same, all of the data in there. So what are they comparing the newly generated tree against in terms of, when they were comparing the newly generated tree to the original tree, what are they looking at?
They're looking to see, maybe it'll be better to look at it pictorially for a second. So that you had an original tree generated from your data set. Then what they're doing by randomly selecting these columns and resampling them, they're saying that in this case here, it didn't matter the order of the columns. Sea lion and seal always ended up being together, clade together, 100% of the time against so it's clading like the original tree 100% of the time. So it does first generate a normal tree, whatever kind of tree, your maximum likelihood or neighbor-driven tree. Then it takes and resamples using the same tree building method and algorithm and whatever models you picked, and tries to resample to see if it's gonna get the same clustering. So it's not a perfect way, but they don't statistically have any other way of testing the trees, and so they say that if in resampling you get the same sequence, same clading or clustering, then that you could say with some confidence that that's true. So again, it's.
I guess I just don't understand what would cause the sequence rearrangement to come up negative.
Well, it's just intuitively I understand what you're asking. Realistically, it does.
Mm-hmm.
So, because intuitively you would think, okay, well, it shouldn't matter, right? If it's lined up well, it shouldn't matter what order each of the columns are.
Mm-hmm.
But it does, and part of it's based on the algorithms you choose. So if you're looking at so even though each column is independent of every other column, some programs have some, like it's looking at third sites. So if the third site is not site 3, then that might have a difference, things like that. That's the only way I can think to explain why you might get a difference.
Okay.
So when would you decide that your tree is no good if you had like several low numbers and a few high numbers? Where do you draw the line?
All you can do is, and this may happen with your data sets, which is fine. All you can do is say, I can say with confidence that these traits these genes are significantly close together. It appears that this clustering, dividing this from this set is accurate, but within this it's kind of messy. In other words, so you can only predict that might be true. That's the issue. And even here, we can't even say with some confidence that the monkey is a good outgroup for this, because we're not necessarily getting, what it probably indicates is that sometimes the monkey is landing up in these areas up here. So when you're discussing trees, and when you see trees in the literature, they can really only say something about the significant tree, significant branching. Does that make sense? Not that, there are people who publish who say, oh, look, the monkey's the best outgroup. And they don't know what they're saying, but.
So bootstrapping doesn't generate a new final tree, it's just used to validate the tree generated based on the criteria you put at the beginning?
Yes.
Okay.
So in Mega, they actually have two tabs. They have the original tree with the bootstrap values on it, and then they have the bootstrap tree, which is really just a condensed tree. What it does is it would condense, I don't know if I have that in here. It would condense the branches that were not, it would collapse them like this. So this is what the bootstrap tree would look like in Mega. This is what the original tree would look like with bootstrap values on it. So this is the tree we want. This is just showing you that there's not a whole lot of significance in these sequences here. Okay, everybody good? All right. So do I need to go through the text part of those slides again, or do you feel comfortable? All right. So this next few slides are just talking about how we can figure out times of change between sequences, and also quickly how to design primers. Okay, so because those are also part of your project. So not all amino acid chains mean the same thing, we know that. And so that's why we use programs like Hammer Blossom, which weight the different types of changes. So it's something to keep in mind. And it's also interesting to think about which is better to look at, the protein or the DNA. So the way we set up Mega now is what's nice about it is we can toggle between the two. It used to be that you actually had to do your protein lineup separately from your amino acid, your nucleotide lineup, and now the programs are overlapped. So you can you're actually making your lineups based off of DNA sequences, but you're going back and forth between your protein sequences and so it helps you kind of figure out some of those silent changes and things like that. And the value in that is that when you're looking at silent changes, it can give you some indication of how much mutation is happening in those genes, because that area there, those sites are free to change if it's a fourfold degenerate amino acid without any selective pressures, right? Because it's not really going to change the amino acid. So it gives you an indication of the rate of change that might be happening in those genes, because they're not being selected against, so they're not being eliminated. So that's where that can be really helpful. And then protein sequences can give you more if there's been functional changes, because if we actually do see a change in the amino acid, what does that do? I mean, if we eliminate a tyrosine, which is a phosphorylated site usually, what does that do to the protein? And if it's still a functional protein, then that might indicate, maybe it conforms just a little differently in the organism or something like that. So we want to make sure that we're looking at open reading frames, which at this point, all of you have open reading frames. All that means is that it appears to have all the signature sites of a real gene. It has a start site, has a stop site, things like that. No stop sites that are premature, things like that. So once we're looking at an open reading frame, we can compare synonymous and non-synonymous changes. And I think I already mentioned that when we were talking about models, but if I didn't, synonymous change just means that the change happens, but it keeps the same amino acid. So that's where we're looking at like the fourfold degeneracy. Non-synonymous changes actually change the amino acid, and I think we can also call those silent versus replacement. So which one is more likely to be selected for or against? And of course, replacement would be the more detrimental. Right? So by looking at those rates of change, we can actually determine times of divergence. And this is something I'm having you do in the lab, and also something you're gonna look at in your project. So the question here is once you've done that, you can figure out times of divergence, and then you can see, do those times of divergence for these genes make sense based off of what we kind of know already based on fossil record of the organisms that you're looking at, okay? So you're gonna have to look that up a little bit for your project, not for the lab, because when you're analyzing that. So in other words, does it look like it's making the most sense? And, that we know, I think the time of divergence between monocots and dicots, for example, it might be something like 500 million years or something like that. So are you getting numbers close to that when you're comparing the two or not? I'm not absolutely sure that's the right number, but anyway, you can look those kinds of things up and determine at least big groups of change. Like if you're looking at vertebrates versus non-vertebrates, you can figure out the time of divergence, or even between mammals and birds. So just keep that in mind as you're looking at your sequences. So here remember the formula to 2RT equals K, or is it the other way around? I can't remember. I think I have it in here. I have that formula in here, because now I'm forgetting that formula. Yeah. So mu equals K times 2T. So 2T mu equals K. So I did have that right in my head. So what I have here is I actually have the mutation rates that have been determined based off of looking at a variety of genes as mutation rates within these organisms. Okay, so this is our mu. So we have, obviously there are the mutation rate for animals for non-synonymous substitutions is a smaller number than for synonymous substitutions, because of course there's pressure on these ones and not on these. And so you can see why that might be. Does everybody? So bookmark this slide, because this is the slide you're going to need for the lab today and for your project. So, and also some of you, I don't think anyone in this class is working on mitochondrial genes, but in the past I've had students work on mitochondrial genes. And so those are the rates of change that we typically find in mitochondria. And so again, these are the rates of change we find in bacteria. I think Lawrence and me are looking at bacteria, in plants, and in animals. Okay. So today in the lab, you're going to be using these. Okay, so I'm going to give you a hint. In the lab, you're going to take a distance matrix. Those distance matrices are based off of looking at K values, right? So the distance matrices could be generated using Jukes-Cantor or Tamura-Ney or something like that. So I'm gonna walk you through that in the lab. Take that distance matrix and paste it into an Excel sheet. Because then all you have to do is put the formula in. And you're not hand calculating every single square. Okay? Hand calculating every single square in the lab would take you a bit of time, but there's only 8 sequences. For your project, you've got 20 sequences. Okay, so everybody know how to put together a formula? If not, I can help you with that in Excel. All right, so here's the formula. Again, that last slide was giving you your mu. You generate your K by generating distance matrices. So of course, your K can be generated for either synonymous or non-synonymous, and you just need to keep track of which one you're doing. And in the lab, I walk you through doing both. So K is the number of substitutions between 2 sequences, and then you can calculate time. And our K values depend on the model of evolution we use. And the other thing to remember is that these distance matrices are what are used to help build the trees. So then you can go back and you can look at your times of divergence between the two, whatever you generate for T, and you can compare it to what you found on the tree to see if it makes any sense, you know, which sequences are. It should, because it's kind of a circular argument, right? So that you're using the same distance matrix to develop time and to do that. Now, the other thing is that make sure you're recognizing that you're using, you know, like 10 to the negative 9, or you know that this is 0.85 times 10 to the negative 9. This is just how we write it when we can't with the subtext. All right, so again, for K, what we would do is count what fraction of each substitution sites can have a synonymous change and how many can give a non-synonymous change. So what the program's doing is it's taking proline, so how many synonymous changes can we have with proline? Angela?
I just said one.
Yeah, so right here, right? So we can have a synonymous change at this site and a non-synonymous change at each of these two sites, okay? So the formula actually takes into account the average number of, in other words, it looks between the two sequences and it looks at how many synonymous changes could happen, and then it compares to how many did happen, right? And that's part of how it generates the K value. So each codon between the sequences is observed for the number of synonymous and non-synonymous changes. Easy when it's one base, but remember there are some that change twice. So if you had CCC change to CCG, that's just one synonymous change. But here we've had a synonymous change and a non-synonymous change, because we've had the second base change too, okay? So you have to figure all this out. So again, you're not gonna have to do this by hand. I just want you to realize what the program's doing. And then you correct them for Jukes or Chimera 2 parameter, are the two most common ones. And in those cases, we're gonna see that you can do Jukes Cantor for synonymous, non-synonymous, or you can do Chimera 2 parameter, synonymous versus non-synonymous. So again, you're looking at the fraction of the possible changes versus the actual changes. So again, it depends on the moment. So this is all done for you. So you don't have to do, I'm not making you do this one out. All right? Everybody clear on that? Okay, so that's another part of the project that we have to have done. Yes?
So your K is only between two sequences, or will it do it for all?
So it's it does pairwise. So it would do it between A and B, A and C, A and D, A and E, B and C, B and D, B and E, right? So you're gonna end up with a matrix, and in each square, it's between two sequences. So you're not getting a single value, in other words, you're getting a single value between each pair.
And then would you calculate the time for each one?
So that's, yes. So then that's why I'm suggesting paste it into an Excel sheet and just apply the formula. Because you'll have mu and you're solving for t. Okay, so then the last thing I want to talk about, I think everyone in here has done PCR, right? Everybody? Yeah, so you know that we're adding primers, and usually someone has to design those primers for the sequences you're looking at. So this is just what I'm trying to show you is that I'm trying to get you to think about that, because we actually have programs that can help you design primers. But you still have to go back and look to see if those are reasonable primers. And often what we do is if we have a gene of interest that hasn't been sequenced yet, is we take old sequences of related organisms, maybe line them up, look for conserved regions, and do the sequencing. And that's part of the reason I was asking Rachel, or Dr. McElpring, the question I asked her about the ribosomal. So it turns out that what you want to do is do exactly what she said she did. They take the conserved regions, because the in-between is not conserved, but then they can find the uniqueness of the gene that they're looking at because of that not conserved, okay? So it's kind of planting that question in your brain so that you would recognize that you're designing primers in conserved regions and then try. So what you'll do is you'll take your MSAs, look for conserved regions, and then you might have to just take 1 or 2 sequences based off of that to try to design your primer on. But you can also design them so that they're a little bit degenerate. And so what happens is that, anyway, I think I actually have that in a slide, so I'll talk about that in a minute. So of course we know, I'm not gonna go through the idea of, I don't know how to do PCR, but the idea is that you design primers that are about 18 to 27 nucleotides in length. And so the longer they are and the more conserved they are, the more accurate it is where it binds. So that's why you want to get long enough sequences. Statistically though, 18 is plenty if you think about the chance of an 18-piece DNA sequence just randomly binding is pretty low. So, but again, we design these in conserved regions because we do want them to bind to our gene of interest, a sequence that might not have been found. And the idea here is when you're designing your primers, you don't want any hairpins. So you don't want any palindromic sequences or any areas where secondary structures can form. And you also usually want a high GC concentration towards the which helps you determine the temperature range, but also towards your 3' end. Okay, so that's the clamp area, that's the area that the polymerase is going to bind, just like in in replication, you know, you've got your polymerase binding, so you want to make sure that's a strong clamp. It's not kind of floating off because it doesn't quite match. The other thing to avoid is primer dimers, and all that means is that if you have your primer here, and if they have reverse sequences on them, they might end up binding together instead of binding to the DNA. So you just have to try to avoid that. And so programs actually do that for you, but you also want to eyeball that because I have yet to find a perfect primer design program because there are certain things that you have to kind of keep in mind. But they do give you a place to start, and there's actually a primer design program embedded within BLAST. So you can take a sequence and it will design different primers for you. And you can start there. You can look at those and see how they look in your lineups to see if they're reasonable or not. Okay. So I want you to think about that. And again, you can have degeneracy, but it increases the fold of the degenerate fold increases the complexity of the cocktail of primers that you're applying. So you don't want too much degeneracy, but you can have some. And again, you can base this off of looking at your consensus sequence. All right, everybody. So the other thing to keep in mind is you're not going to want to build primers, and I will look at this on where introns are. This is part of the reason why I want you to know where the introns are in certain sequences and find out whether they're in the same place or not. Because if you have a coding sequence, you're building your primer off your MSA, which right now is based off of coding sequence, and you have a, you have a sequence here, and then you have your and in other words, if your intron's here and you build a primer here, it's never going to work on genomic DNA because it's only going to bind the first 3 bases, and then everything, it's not going to find a hybridization place. So it's important to know where your introns are. And if you want to make my life easy when I'm grading these, you will point out where your introns are in your sequence and say, I did not put my primer there. But sometimes going right up against an intron is actually a good place because there's often conservation there. It turns out that in a lot of cases, intron placement is the same. And you can think about it because of if the gene copy is old, it maintains that intron throughout. So just keep that in mind when you're designing your primers. Okay? Did everybody understand why? Yeah. That's part of the reason why I want you to look at, had you pull out some genomic sequences so that you can see. Because the idea would be that you would do your primers during, with genomic DNA. All right, so you have your GC clamps at the 3' end. You're looking for, to eliminate primer dimers. You could have some degeneracy. And you base this off your MSA consensus sequence, and then you note where your introns are. And then, of course, so the other thing you can do is you can actually nest your primers. And I think that's what I have you do. I want you to think about nesting primers. And the reason to do that is if you're looking at something that you don't have high copy number of, nesting it will give you more. So what happens is you do one set of PCR with the furthest out primer set that you've designed. That will give you, after that PCR is done, you can reset up that PCR, taking a small portion of that and applying a different set of primers that are in a little bit, okay? So they can have a little bit of overlap with your outside primers if you're trying to optimize as much as you can get, but obviously they can't be exactly, because the ends don't always get maintained in PCR. All right. So that's the goal of the primer, the nested primer idea. So in your case, you could probably just figure out if you were going to use your promoter region based off the couple that really are pretty similar,
Okay.
rather than your entire MSA, because it's the rest of it's.
Good, I was worried about that.
Yeah, yeah. And in your case, like what they often do with promoter regions is they use something called 5' race, where they've got something that's designed against the gene they know the sequence of, and then they have a cocktail of random primers that are pretty typical in promoter regions to try to get that in. Okay. So a good set of primers include all of the following except, here's your question, one of the day. So hopefully you put 2. You don't need a 5' GC clamp. In fact, sometimes that end can be a little more degenerate. All right, so the next question, the next slide is actually a quick write question, which I'm not sure I actually put on quick write. So, I guess before I got the right question. So what I'm going to talk about now is not in your book, and I've kind of simplified it based off of what I think you want to explore in terms of your own project. So we're going to be talking about systems biology, and part of the reason why is, and actually it was Dr. McElcrane mentioned this too. It's the idea that our genes are not in isolation, right? They work usually in concert with other genes and metabolic processes and things like that. So we're going to look at how we can determine whether our gene is part of some of those, based off of known systems. And in fact, one of the databases I'm going to show you is KEGG, which she talked about too. And there's a lot that goes into systems biology, and it is kind of a newer field of bioinformatics now. And you can get degrees and actually study those kind of processes and build models for how this might work. So there is a little bit of modeling process. We're going to talk a little bit about the model types. I'm not gonna spend a lot of time on the mathematical descriptions of those, but obviously recognizing that that's behind the scenes, where they're kind of figuring out things like the kinetic energy of the chemical reactions and things like that in those, okay? So, just, I'll show you that. So, what happened to my, oh, see, I went too fast, that's why. All right, so the quick right question is, what does systems biology mean to you? What do you think that means? And I'm gonna put that on the quick right. Obviously, just pick the next column. Okay, so I just realized there's, a third of our class is gone today, so it's like, what? Oh, yeah, that's right. The first 3 people in this class are gone. All right. So I think all of you get the general idea of systems biology, and again, it's kind of something that more people are focused on now, and in fact, it's my interest in looking at biosynthetic pathways. Because the other thing to think about it is, how did those pathways actually evolve? What allowed for the flexibility of proteins that had to work together actually come into being? And so was it that we built these pathways sequentially, so you started here, this for a while was what organisms used, and then they added a piece, and understanding those kinds of questions and those kinds of directions. And some of that can be explained by gene family duplication. So a lot of times genes in the upper part of a pathway can be used in a number of pathways, so there's a number of branches. Because most pathways are not linear, they're kind of a web of pathways. So that's one explanation. Another explanation might be that there's a chunk of a pathway here and a chunk here, and then they merged, and things like that. So it's an interesting question, and we don't really know the answers to those kinds of things. So it's kind of a interesting thing to look at least from my perspective. Therefore, I am doing research on it, or trying to do some research, contribute knowledge to it. So, so we just did that. Okay, so because it's alright, I wanted to keep this short. I just want you to look at this. I'm not going to ask you to quick write this, but let's just look at this together. Oh, so you know when you have these on your slides, by the way, I don't know why, but it no longer works where it goes directly to. But if you click on it, you can get to the hyperlink. Click on the hyperlink, I guess I should say. You can get to the hyperlink, you can edit it, and then you just, what I do is I control copy and I put it into, but I think I actually have this one opened, because it was a good idea, it's right here. Okay, so this, this is a article that was written in 2013 about bioengineering kidneys. And so part of the problem with trying to build organs has been this issue of, it's not, there's all kinds of things that are involved in that organ being maintained in a body system, which is why we have problems doing transplants, right? Because it's not, you can't just say, oh, I need a new kidney today, I'm gonna get one from my sister or my brother or something like that. It's not as easy as that. And so they have to figure out what it is that has to match and things like that. And so this one is pretty cool. It's a pretty cool story. I wonder how long yeah, let's just watch the video because then it'll give you some ideas. It's kind of cool.
Frankenstein were real and alive today, he'd probably want to stop by the Massachusetts General Hospital in Boston. Here, scientists are harvesting dead organs and returning them to life. But instead of giant green monsters, these researchers are creating hearts, lungs, limbs, and kidneys that could one day be used to replace failing organs in people. Reporting in Nature Medicine, Harold Ott and his team describe their vision of the future and how they created a functional, transplantable kidney in rats.
What we try to do here is to regenerate organs for transplantation, organs for patients with end-stage organ failure.
Using donor organs that would not normally be suitable for transplantation, Ott's team reanimates them by first washing away native cells with a soapy solution. What's left behind is a natural scaffold of structural proteins that can be repopulated with stem cells taken from a patient.
What's fascinating is that you're able to, with that process, preserve the blueprint of the organ, but it's completely washed empty. It's a mere, a shadow of the original tissue. Now the major hurdle here was to get the cells, the viable building blocks, back into the right spots within that organ. That we did through creating literally a pressure gradient across the kidneys, so we're able to suck these cells into the tissue, into the kidneys, into the right spots.
The organs were placed into chambers that simulate the environment of a body, complete with oxygen and nutrients. After a few days of growth, something remarkable happens.
The tissue became functional, so these kidneys started to make rudimentary urine. In the last step of this experiment, as the ultimate proof of principle, we were then able to take these kidneys from culture and transplant them in an animal model. So we took that final step to take the kidney out of the bioreactor and then treat it like a transplant organ, like a donor organ, and replace the native kidney in an animal with this engineered kidney.
But it's not always that easy.
We had to figure out how much vacuum can be applied to the kidney in order to provide just the right amount of tissue gradient to deliver the cells, but not to damage the matrix. Of course, if more vacuum is applied, we had quite a few kidneys blow up in a jar.
Hiccups aside, laboratory-grown organs appear to be closer and closer to fulfilling their tremendous promise.
The reason why I'm most excited about this recent publication is because it shows the platform character of this technology. The technology can be applied to any tissue or any organ that can be perfused by its own vascular system. Ultimately, this is a very translational kind of research. It's very much focused on driving this all the way down to the bedside, getting this to the patient.
So that's kind of a cool way that systems biology was used to help model an organ. So and he sort of alluded to a lot of the things that they had to figure out. They had to figure out just how to maintain that scaffold, how to get rid of everything else so that it doesn't get rejected by the organism, things like that. So anyway, just want to say that. So, I'll let you read this. So this kind of gives you the kind of the umbrella of how we are thinking about systems biology, and the fact that it is more than just the elementary parts or the small parts, and a lot of you mentioned that in your definition. Something to keep in mind. So as we think about how system biology has developed, we can see that we've looked at environmental doing actual experiments, and then we've applied these models to those to see if what we're doing is matching up. And so we're going to talk a little bit about that in the approach that we can take. So the concepts that we use obviously are things like genomics, transcriptomics, all the way down to network modeling. So again, nothing works in isolation, so what should be considered? So it's more than the, I don't know why this is doing this to me, but I can't read can't read my slides on that anymore. It's not big enough, but, so here's an example. It's more than the sum of its parts. So if we think about something like how calcium works in our body for bone growth, we know that there's things that have to happen before the calcium even gets to the bones, including how do we get calcium into our bodies. It needs to be absorbed through the intestines, and how some of that gets excreted, how it interacts with hormones. So again, all of this is looking at the whole system, so it can give us insight into how calcium is related to bone growth and understanding all of that. So it's more than just a simple idea of, oh, bones need calcium to grow. So when we're modeling this, we have to think about all of the digestive parts and the hormonal parts to getting calcium to the bones, and how the importance of that. So there is an interconnectedness, and the other thing is often you can go more than one direction. So in some cases, you can actually reverse some of these pathways. And in some cases you can't. And so understanding what you can and can't do is important. So an example could be this made up figure from a book of a train and it's a transportation map. Okay, so you can see first thought squares. But you can see that you can have different ways to get to certain places based on the path you take, okay? And so you can have different ways to get to Sweatshop Park, for example, and the solid lines are supposed to indicate, I think, trains, and then the dotted lines indicate other modes of travel, so, and also pedestrian ways of traveling. So you can see that, first of all, you have to figure out which kind of transportation you're going to take, and then secondly, what path you're going to take. Okay, so this is just an analogy that the book was using. This is also something to keep in mind is that this is from yet another book where we have to think about what space we're working in and what's true about each space. So for example, we have a gene space, a protein space, but a lot of times some of these proteins that are built then come back and work on another gene because they're transcription factors, And then from there, we go into a metabolic space, and which again could ultimately have a loop back to what's happening with gene expression. So if we look at, again, I have, actually in notes, I tried to paste, you know, the notes at the bottom of the slides. I tried to paste the website, the most up-to-date website. So if we go back, I have KEGG open already. So here's what the KEGG database looks like. And you can search for anything. So if I do gluconeogenesis, so you can see that you generate a page that looks like this. And so there's different pieces that you can use, including just looking at the genes that are assigned to this pathway. But if you look at the pathway itself, I think you have to do this on the map. This shows you how interconnected all of this is, okay? And these numbers here represent the enzymes that are interacting at this. And these are you can put these numbers in and it'll call up the actual enzyme for you. Those are the accession numbers for these enzymes. And so you can see that there's a number of different directions you could go, starting with starch and sucrose as you move through this pathway. And then what often happens, these ones that are more circular like this, are actually pathways within themselves. Okay, so so there's another set of pathways that are here, okay? And then you can, anyway, you get the idea that they're very complex. And this is a way that you can look at it. And so, you're looking at a gene of interest, this also gives you some of the diseases. So glycogen storage, if you click on that, it's giving you the idea of what is happening with that disease and what it ends up doing, and then it shows you, I don't know if it'll highlight where in the pathway it was, it might. So, yeah, so it's highlighting in the pathway where change happens if you have this syndrome, okay, so if you have and there's probably a key to whether it's the red or the black. And it might be that it upregulates, downregulates those enzymes or things like that. So usually the color represents that, but I actually don't know what the legend is for this. All right, so that's one. And so this is actually a very common database for most of you could use that database because it has both plant and animal systems in it, so you can look to see if your gene's in there. And the way you can do it, I think you can actually search a gene and see which pathways it's involved in on that one. So the other one is this EcoSys. This one actually gives you a more, a smaller pathway, like a sub-pathway, giving you things of directions of the reactions, and also emphasizes the control. So that shouldn't say shoe, it should say show. Control the pathway. So again, this is the site here, but again, I think I have it opened. Good. And this one is based off of E. coli, so it's giving you the E. coli site. So again, I just searched glycolysis in this case, and you can see the direction, and you can have more or less detail. So if you go more detail, it's going to give you some of the other regulatory other gene products and things like that are involved. So this is another program that can be used to kind of see where your genes land. And so Lauren might use this because she's looking at a bacterial gene. And then, and this is, right back to the slides. here's another example, and in this case here, the solid lines are enzymatic proteins involved in the pathway system. The dotted lines are the transcription factor proteins that turn on these genes ultimately to be expressed. Okay, and so you can see these are the protein-protein interactions, all these solid lines leading to there, and you can see that in certain places they've highlighted where there's lots of directions you can go once you get to that gene, which I can't read because my eyes are big. But makes sense. So again, it's all very complex. So how do you model this? Well, there's actually, as I showed you in that first slide where I was giving you the flow chart of what's happening, we can look at this either as a top-down, middle-out, or bottom-up. And it depends upon whether you have experimental data or you have modeling data for these different things, and then you can use that. And this is just showing you some of the types of models that are built based off of these things. So in bottom-up, the constraint-based models and models of reaction kinetics, and then top-down are statistical models and pathway analysis models, etc. So a bottom-up example might be they start with data on black eye. So we know a lot about black eye. It's been studied extensively, experimentally, so we can use it to figure out a model of how it works with it interacts. So here, so they designed a model where they were looking at promoter regions of black eye to see how they worked in relationship to the fluorescent green proteins. And what you see here is that one of these is the model, which is the blue line, and then the red line is from actual experimental data. So that the model they built, or to describe this, is actually pretty close to what they found experimentally. So that would be considered a bottom-up approach. A top-down example, you start with a pathway, an observed pathway, and then figure out how to model that. So, for example, tumor invasion, where they might actually model how they think the proteins are working. So here's an example of a computer simulation of how a tumor might grow. So it's growing for a few days, for 45 days without a whole lot, and then something happens where there's a few kind of what you call aggressive cells that then really take over, and the tumor starts to really grow. So this is a model of what they're thinking is happening, and then they take that model and they see if they can figure out if that's truly what's happening. So it's the other way around. So they build the model first and then look at the data. Okay, so middle out would you take both types of data and expand, and it's the most often used. It's the one where we actually take both modeling and experimental data and then start expanding from that. Okay, so this heart model is kind of pretty cool. They use real data and modeling data to try to figure out how tissue gets built and repaired in the heart, in this example. So that's just what that showed. So the question is, which parameters should you use for these models? And some of the things you have to keep in mind are, biologically, we have to think about time in development, time of when evolutionarily, and also what species we're actually looking at. So there can be some constants and some not. And then there's also, we need to consider feedback, and feedback can be both positive and negative. So some of the parameters for the model might be that you have these certain molecules, but there's certain things that have to happen in order for molecule A to interact with molecule B, which then can produce molecule C, or that kind of thing. So all of these have to be taken into consideration, and we also need to think about. So we need to think about what the output and input is into each of these systems, and we also can begin to think about feed-forward control versus feedback control, where reactions are based on the input, and then the more precise is the feedback control, which is usually that the desired output and actual output are compensate, and this can happen both positively and negatively, right? So we can think about, or you can think simply about the lac operon system. Once we've digested, it gets put into work when they're lacking glucose, and so then they use lactose, and then once glucose comes back again, it'll turn on or off the system so that it's, understanding all of that. And of course, that's one of the first systems that was studied extensively. So here's an interaction of negative and positive in apoptosis, right? So there's certain things that turn on cascades of reactions in apoptosis and other things that turn off. And so in this case here, when you have arrows, that means it's positively controlling that. When you have this is negatively controlling that. So in other words, this has a negative feedback loop onto this, which stops this, which then means it can't go there, that kind of thing. And so, and of course, it's not simple, right? It's not, there's a lot that's going on and what has to happen in order for apoptosis to occur. And here we have the, and they're also showing some of the organelles that all of this is working. Okay, any questions about that? So, and this is a, that, it's the same thing, same kind of model. To me, it's a little clearer because we can actually read what's going on. So it's not as cartoony, but actually in some ways it might be more informative, right? Showing us the positive control and the negative control being positive, and the red being negative control. And this is all leading to cell death. What happens in order for cell death to occur or not occur? So there's different equations that can be used for this. So even though I'm not going to go into the detail of this, but recognize that we can differentiate rates forwards and backwards, and they might be, it might be simple like this, but then again, to get C might actually end up having a more complex component to it. And so we have to think about the involvement of all this, and again, most of these pathways are not linear, so this can be used to help build the models. So here's an example of EGFR, and here's so again, we've got the cartoon of what's happening here, and then we have the results of the model based off over time. This is a summation of what we're seeing. So the other thing we need to think about is the robustness. So how do proteins actually respond to changes in the environment? Think about something like heat shock proteins, things like that, and how those actually help turn on or off different genes. But there needs to be a change in the environment for those to occur. So you can think about Legos, what's needed to put together, and there's actually what's the important part, what's not so important. And so here, if you think about the modules here, so what's important is that you've got these buttons in order for the interconnectedness to occur. What's not so important is maybe like this, or the color of these, or something like that. So that a lot of times you have to look closely at the components as to what part of the component's important and what part's not. So again, and then you're looking for key points in the system. Okay. So if we look here at glycolysis, you can see that these are pathways within pathways that ultimately lead to the production of ATP. But you can see that that there are pieces that get you to a product, and then at this point, this product can actually go in several directions from there. So this is a simple example because most of you are very familiar with this. So what else has to happen? We need to think about redundancy, and glycolysis has a lot of redundancy in it. There's gene families, and that could be also the idea of gene families, which is what I was talking about originally. some systems are irreversible, something like the cell cycle. So you can't get back into the cell cycle until DNA replication occurs, for example. So the model needs to include whether there's redundancy or not. And so again, we can look at in KEGG, for example, we can look at steroid degradation and see the result of that. So let's go back here. Let's go back to the beginning. I don't know if I spelled steroid right. No, I did. And it's not going to correct it like Google does. Steroid. Okay, so then again, we can look at these pathways to see how the results of what's going on here and how degradation of a steroid can happen and the different enzymes that are involved in that. And whether there's any kind of feedback loops, which it looks like there can be some alternative routes to getting certain places. Okay, so that's just really the goal there. So that really is just showing you some examples of websites to look at. And so there's links to Mini and what I was going to have you do there. Again, it's not going to probably let me look at it. Was just I had a survey where you could look at those, but due to the fact that I went a little over, maybe we'll do that. You can do your own, just look at some of those sites and fill in that survey. I'll give you a couple points towards your homework for that. So I think you have to go into, again, just go into hyperlink. Put it hyperlink, and you should be able to get it. I'll try to put those on the bottom too. So it should give you. So the idea is that you try a couple of different programs, and so just name 3 things that the website provided for looking at this, and would it be helpful for you? Okay. So look at, I'll say you should look at 3 websites. Just skim through 3 of them. So that'll be a part of your homework. And then we can talk about it later. Especially if you find a one that seems to be useful for other people. So, and then this one should be a a page that has links on it. Yeah, so here's, some of these might be dead, so you just have to look, because I don't remember the last time this, it probably, yeah, this was, well, it says it was updated in 2018, so they might be good. So just randomly pick a few different ones and see what might be of use, because eventually you're going to have to do this with your project. All right, so rest of the time is yours.